Good morning, all of you. And this session, I'll be extending our ongoing discussion to the homology modeling and threading algorithms. Here, as we have been discussing in the last lecture, homology modeling implies that we're using the homology concept here, present in the already solved protein structures. So we are using them as templates to model our sequences whose structure is unknown, which we are calling them as targets normally. Okay, so we are building the target template alignment and then we are building their model. Okay, using several different tools are available like a modeler, which is the widely used tool. Likewise is the threading algorithm, where what is the concept? Like for these template information, rather than taking one or two or three templates as used normally in the homology modeling, threading believes that threading is based on the concept that the folds are well conserved. Means supersecondary structures which we just discussed in the last lecture are well conserved in the like different protein structures, PDBs. So it keep on uh, first parsing the sequence into different folds, different functionally conserved substructures, and then it keep on taking these substructures from different PDB sources, and then it lastly links them together, seals them all together like annealing. Okay, so there are several different algorithms like simulated annealing and others. Okay, so it is linking all of them together then to minimize, and through energy minimization it builds a single model. So whenever sequence similarity between the target and template is greater than 30%, homology modeling gives a reasonable model. Okay, but when the sequence similarity between target and template goes like between 10 to 30% middle range, okay, then threading works out. Because threading, rather than considering the entire sequence into an alignment and building an alignment for the entire target template, it breaks the target into small segments. So the sequence similarity between the individual substructure and the template like domain substantially increases in similarity. So it directly adopts that domain topology here. Okay, so the accuracy substantially increases. So threading is able to build a very good model in the cases when this homology modeling fails for the low sequence similarity cases. And for all these models, I'm using several different scoring methods, like as we discussed Ramchandran plot here last class. Okay, we'll be extending our discussion to further more scores like solvent accessibility and others. Okay, see like templates based modeling or free modeling. Free modeling includes ab initio and this threading methods. Template based modeling method using template TBM homology modeling. This is the way the algorithms have been categorized. So this categorization has been done by CASP, okay, which is the biannual test taken every 2 years, termed as critical assessment of structure prediction. Here, what is done is the researchers what they are doing. Few researchers who are organizing this test, what they are doing is whatever the protein structures which are just going to be released in the PDB database, they just stop the release. Okay, and what is done is that how do they select these specific targets for test? These targets should be something new, that which cannot be easily solved, okay, easily taken up. So they're easy modeling target, okay, which are difficult targets like that, they categorize. So easy means whose templates can be easily available. So for sequences whose templates are easily available, they put that in that category. So for targets whose templates are not at all there, means less than 10% similarity are there, they put that in that category. Then now, like in discussion with the persons who have solved the structures, they take the PDB data and they keep them with that. Okay, they keep it with them. They don't release it in the PDB, which is then released after the exam, after the test. So these protein sequences are given to all, like all the persons who are taking the test. This test happens every 2 years globally. So several teams, like even across India also, some people participated. So what happens? Like every team is allowed to submit at least 5 models. Okay, 5 models, 5 of their best model for every target, whichever they are participating. Okay, like that. So whichever the team is that confident that these 5 models are best, they can put it there. So lastly, as per the final answer which they are already having, they keep on ranking the solutions. So whichever the person win that, whose the maximum most of the answers are correct. Okay, by considering RMSD like TM score and GDT score, global displacement test score, and like that, they announce the winner. Okay. So see, as we have been discussing that the structure is conserved in sequence. Structure of a new protein derived using protein structure as templates. It is evident for the evolution. We have already been discussing all these concepts. So see, like we are now discussing the further steps. So homology modeling implies that what are the steps used? Template selection, building the target template alignment, model building, model refinement, and model evaluation. Okay. So if some people find that model is not good, they can go for the refinement step or doing it iteratively until the like alignment the score improves or store like assessment score stops in further improvement. Okay. So what is the basic idea here? The basic idea is see sequence similarity for a protein sequence is higher against the experimentally solved protein structures. Means protein structures are much more conserved. When one disposes of a sequence for proteins with greater than 30% similarity with query means target, means when the target template identity or the target template similarity is at least 30%, I can make a very good model. But if the similarity goes beyond below 25%, then the like template based modeling cannot simply work out. Okay, we need to normally take threading to improve the modeling accuracy. See? Several different examples have been there in the past, which have been well published. See, this is a triose phosphate isomerase, temporal domain. See, the helices are well arranged here. See, when I'm building it for this triose phosphate isomerase for a template with a 44.7% sequence similarity, I'm getting a model with an RMSD of 0.95 Armstrong against its actual answer. See, the model is like light green color here, and the template is the blue. See, exactly overlapping, and the see ligand, the native ligand is exactly fitting at the same place. Means it's very accurate structure. So see, that's the advantage. So when the sequence similarity is high. So now see what is the major requirement for increasing the accuracy. We have been discussing sequence similarity. So sequence similarity plays a substantial role here in the modeling methodologies. See the is dependent on the sequence similarity. See, I've taken it from different research papers to make this videos much more interesting and much more ahead of the part. Okay, because this course is advanced bioinformatics. See, percent structural overlap. Now you understand that if the sequence similarity increases, uh, the structural overlap or the like a more uh, structural correctness between the target model or the generated target model against its actual answer or against its template will be substantially high if the sequence similarity increases. See, sequence identity, I'm calling it a sequence identity here. See, more it is high, it's going here, see. But when it's reaching 30% here, see, I'm getting substantial structural overlap. And structural overlap is constantly increasing. But when the sequence identity here shown is 60%, it's almost close to 100%. See, range of residues, there is now sequence similarity in percentage identical residues. So when 100% sequence similarity is there, I get the speed. See, the key limiting factor for the model building is speed. It's quickly building out. Most of the time, it's same model. I'm unable to impart the additional key variations in the model which is supposed to be there for any model. See, it's working out cool, like speed. See, likewise is the quality here between 50 to 75%. When it goes from 25 to 50%, it becomes alignment. The alignment error comes up. But when it goes further below, okay, the alignment error means the gaps. I'm unable to put the gaps properly. Okay, wherein HMM based methods comes up. So which considers the predicted secondary structure of the target sequence. So they predict the secondary structure of the target and then properly adjust the gaps, adjust the sequence gaps. Okay. So when it goes further below 0 to 25%, I'm unable to detect the templates, which normally happens. So the homology modeling fails, but threading works out because threading already has the like domain database, well conserved database domains. It already has everything, so it uses that and work it out. So now see to we'll be discussing the steps of the homology modeling in more detail now. So rather than considering the templates as one step, I'm calling the search or template search and template selection as two different steps. Well, they are actually two different steps. Okay, but we normally call that as template search and template selection together. Okay, rather than mentioning them explicitly. Several books do it. Okay, so but how do you search templates? We use blast, we use HMM based methods. Okay, but understand that whenever the sequence length is small, you can use any method. Answer will be correct only always. You'll get similar results. But when the sequence length is substantially large, where more number of domains are there, you need, I personally recommend HMM methods. Okay. So see the step 2 means searching the templates. How to search template? So see the purpose of predictor model. We have already discussed all this last class, so I won't repeat it again. See purpose of predictor model, sequence similarity, phylogeny, resolution, e-value. These factors play a substantial role. Okay, in searching the like commendable template. See for now, like step 3, see we are building that alignment. I have just shown the representative alignment here to make you understand. See, understand that if it is more gaps, more number of gaps, multiple times gaps are opened, that is long. It should not be there. The modeling accuracy will be decreased. Moreover, if you are opening the gaps less number of times, no issue. That's okay, advisable. But you should not extend the gap substantially. That will further deteriorate the overall modeling accuracy. Okay. See now step 4. Step 4, what you do, like whichever the parts which is copied. Okay. See here, see the alignment. This is match portion template A, like match portion template B. This is the gap. Whichever the parts which is copied, next process model building. In model building, any tool what they do is they copy the C alpha backbone. C alpha backbone means N C alpha CO. leaving the side chain and the hydrogen atoms. Okay, excluding these things, excluding side chains and hydrogen atoms, if I just take NC alpha CO, that is C alpha backbone, if I take copy these coordinates from the templates and then put it here, means we need to transpose them onto a same part. Okay, so normally what they do, they keep them together, they transpose them in the single like basis lattice. Okay, so that they can be transported in the same shell. Okay, see here, like likewise template B. So normally it's computed as per origin. So template B here, see. So these two, when they transpose, they are brought closer. We can always link them together. For this gap region, we already know. Suppose ACC is there. I know the structure of alanine. I know the structure of cysteine. So what I can do? I can put ACC backbone, seal these two gaps here by making the peptide bonds. Then what will I do? Like that, I can build the entire part. Template A topology, template B topology, in between ACC topology, wherein I've already incorporated the ACC backbone. Okay, now what is left? Only the side chains. So then I can add the side chains as I want. So depending on the side chain automers, like that I can do. And seal up the protein, make the model building complete. Okay, like that. So it is done by energy refinement. If the clashes comes up, because normally the clashes do come up, because several atoms come very close, so you need to properly place them. Remember, energetic relaxation or energy refinement to minimize the clashes. So to alleviate the number of clashes, we need to do all this. Okay. So all this is done by lastly model evaluation. Okay. So lastly we call it as near native model. If the sequence similarity was earlier was substantially decent, we can call it as near native model because it's very close to the actual answer. So like see this part. I've taken it from my own book chapter. Okay, which was published in 2014. You can see it there. See, you can search it on Google and then work it out through Google Scholar. See, if you can find out reliable templates, yes. Select the closest templates, build the target template alignment, do model building, model evaluation. Are you satisfied with the modeling accuracy? I told you, no, we can always iterate. So if you are saying yes, stop it here. You got the model. If you say no, some loop, some like a linker segment or for loop segment which is covering the gap region is not properly modeled. It is coming very close to the like secondary structures. When clashes may be there, I need to still minimize it. Okay. See, like again go for selection of closest templates. Again change the alignment to again do model building like that. Or I can do simply model refinement. If it's marginal deviation, do only model refinement and model evaluation. Again, come up here. If it's substantial variation is there, go here. Okay, but this is the case of like homology modeling or threading. This part. But if you cannot find reliable templates, no templates at all. Can you find reliable aligned folds in the available templates? Means the domains, well conserved domains or the folds. If you say yes, mostly it will be yes only. Okay, for the sequences, okay, it will be yes only because they're made up of domains. So then you take up these domains, perform threading. Okay, then again come up here model building, model evaluation. Okay, like that. Now see like a problem is model. This I already told you last time, but now see here 0.56%, 0.56 Armstrong is the like RMSD level, which is very less. Means everything has gone properly, well, good, well, and well properly it has gone. Means the more protein axis was 3 point something, the RMSD was 3.5. Okay, it was not so good, but so this time it is 0.56. Means templates were properly done, properly selected, like alignment was properly made, model building has gone properly, refinement has been done properly, everything is done properly. So there is no possible errors here. Okay, no possible inaccurate gap here, no inaccurate gap placement or extension has been present here. It's been incorporated here. Okay, so the model is proper. What's the answer here? Ab initio modeling method is helpful when sequence similarity of the target protein is significantly low against the templates. Sequence similarity of the target protein is significantly high against the templates. Both A and B, it depends on the protein. You remember, if I told you correctly, if you remember, ab initio means whenever we are using ab initio, sequence similarity against the templates is very low. It's significantly low. Means it's something which cannot be built by homology modeling or threading. We are only left with ab initio. So if A is the right answer. Why homology modeling is better than other methods of structure prediction? Its accuracy is very high. The first one is of course true because it is dependent on the solid structures. We are not building anything right from scratch, just like ab initio modeling. We are just taking the data from the known structures, which are already correct. Understand? It can always allow us build a protein model. It can always allow us build a protein model. Yes. So, does it always allow us to build a protein model? Yes or no? No. If the target template sequence similarity is less, it will fail. First is the right answer. Okay. Is homology modeling method always applicable? This I told you. True or false, always false. Think the role of target template sequence similarity on the accuracy of predicted protein model. If the alignment accuracy is high, what will I get? I'll get a fairly very good alignment. Okay. I'll get a very good alignment. Because of very good alignment, I'll be able to extract the coordinate details properly from the templates onto the model. Everything will be done properly to result in a highly accurate model lastly. Okay. Now we are reaching the threading part. So threading method, as we all know, is the threading like the strategy when we are linking the protein domains, well conserved protein domains, and then we are linking them together to build a protein model. Okay. Once the alignment is made, a model of the target protein is built by transferring the structural information of the line templates onto the protein sequence. This involves replacing the amino acids in the template sequence, the corresponding amino acids with the target. So as I told you, like we are just copying the C alpha backbone. Side chain we are placing later. That's what it means. Okay. Side chains we are placing later. Okay. Structure is better conserved than sequences, and only a few unique like structure folds exist in nature. Only a few thousand. Means in all the protein structures, it has been well published that like only a few thousand protein folds exist in nature. Means like always in all the organisms, in all of their proteins, like these folds are juggled up. Nature do juggling of these folds in different patterns to create new functions. Okay, they are always connected with the varied loops, loop linkers of diverse lengths. Okay, whose sequences keep on changing. Domains, these domains may have got a different sequence, but their structure will be same. Structure is well conserved. Protein structures are often composed of independently folding, structurally independent and tertiary structure units. within the same protein chain called domains. Their protein folding, while the protein folding goes there, domain itself fold on its own and function on its own. But their function is in link with the overall protein function. Okay. Domain have a specific function in a protein. Every protein domain has a fold, which refers to a unique arrangement of the secondary structure elements. I just told you, I have been telling that. See? Okay, motives are part of big domain and are you unable to fold independently and function independently. Okay. Alright, so see like just as an example I was showing here P53 tumor suppressor gene. So see 1 to 60 here is activation domain within the overall length of 393. 100 to 300 is a separate unit which is specific for sequence specific DNA binding domain. Okay, to interact with the DNA. Then followed by 324 to 355th amino acid is the tetramerization domain. Okay, which forms a tetrameric unit. 363 to 393 is non-specific DNA binding domain. So these four segments are there. Whenever nature needs it again, it copies this part again there. Whenever such function need to be done. Like we do in the house. Correct? So this is the domain part. See? So domains are the fundamental units of structural classification. So see, so see here again, different role in different orientation of secondary structures means different protein functions. See here, same parts I'm using. First one is the beta alpha 8-fold of Timberal. 1XQ7. I have given the PDB also. You can go and check. See this kind of structures. Alpha helices are shown in orange, beta strands are shown in purple here, and loop regions, loop and turn regions are shown in green here. And I'm using the Rossmann part, Rossmann fold. Beta alpha only, beta alpha beta. See? Beta followed by alpha followed by beta, separate, separate. Okay. So see, it's two fold of malate dehydrogenase. Here what happens? Parallel sheets are like present inside, full outside surrounded by helices. Okay, see here. Outside the helices are there. These are sheets inside. That is a specific beauty of beta alpha beta. Okay. See. Here the terminal catalytic domains are there. See. 1 EMD. Now see zinc finger part. Beta alpha beta. See. Similar part. These helices and sheets are used same way but in different orientation, different strategies. So see, it giving different folds, different functions. See, so different folds, different orientation of secondary structures, different function. See, yet another example. So that you will understand better. See, DNA binding domain. This protein was modeled using threading. It had three domains. I've taken it from the CASP structure, CASP predicted solution only, from a research paper. See, it was very big domain, more than 527, more than nearly 600 amino acids. It was 111 to 116 was small part separate DNA binding domain. So DNA binding domain I know the structure. I just shown you. I can take the structure. This is the link region 116 to 195. See this dot 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 dot. This is the loop region. Followed by predicted ATPase domain. That part. Which I'm copying from one Z60B chain. 9e 1.9 into e to the power minus 22. That was the value of comparison. See, all these are doing separate comparison in the PDB by using HMM and then worked out. So I'm taking this part from 2 Q and A. This is 1Z60B chain, then 1WHB A chain. I'm copying this template domain information. See, TPR domain. Then I will lastly combine it here. See, this is a small domain here. Considering there. See, it's end to end, end to end, end to end. I'm sealing it to make a complete one model. So see, if the fold you understand that so protein is aligned with the library of folds as to best so as to earlier described methods and high scoring matches for each segment are considered for threading. Here see how many folds are existing? 4. Here 1, 2, 3, 4, 5. Here only 3 are there. When I want to build a model using threading, it can give me 60 combinations. I understand. So that is normally done in threading. So whichever domain is able to pack properly, I'm able to link it properly. That is only selected. How? Based on the energy. Based on the energy, overall energy of the protein model should be the lowest. I know because protein structures always exist at the lowest energy conformation. I know it already. Okay. Method will fail if the protein sequence does not belong to any fold in the library. Threading fails there. I'm unable to retrieve any fold from the PDB database or it's some null fold. See? Like that. Now tell me this part. Which of the following statements is true regarding threading methodology in protein structure prediction? Threading methods use experimental data to directly determine the 3D structures of a protein. Is of course there. But do they directly use it? No. Threading methods rely solely on sequence alignment to predict structures. Threading method in template selection threading methodology is based on low sequence similarity with the target protein. Threading methods are evaluated using scoring functions and energy potentials to assess their quality. The lastly is always there. See, I'm always using the last part. Okay. Energy is always used. So that last one makes more sense here. In protein threading, the process of aligning the target protein sequence onto a template structure involves comparing the target protein sequence with the database of known protein structures. I'll compare it with the domains. Okay. But yes, I cannot discard the first one. But which one is more correct? Replacing amino acids in the template structure with the corresponding amino acids with the target. I do it everywhere. I am copying the same C alpha backbone always. Okay, assessing the quality of the resulting model onto like using scoring functions and energy potentials. I do it always. Okay, refining the general model to molecular dynamics. For as I'm asking the aligning part here, last three part do not talk anything about alignment. Only the first one does. First one seems to be more correct. Okay. Is threading modeling method always applicable? No. Only when the sequence similarity between the target and template goes like nearly from 10 to 25%, it is more applicable. Think the role of targetability sequence similarity on the accuracy of protein model. Yes, it's always there. Okay, even in the domain part, I need to take the domain from somewhere. So alignment do make some sense. Okay. So now towards the end of this session, we are discussing the last part. Here, what we'll be discussing is how the like few mostly used like protein structure prediction algorithms. Here, what has been done is right from very history, like the Quebec Center was there. Okay, so what they did they developed a dictionary of secondary structure of proteins, like uniformly assigning the secondary structures. So different scientists have assigned incorrect secondary structures in the PDB. So how do you read the PDB properly in a uniform manner? So for that purpose, the DSSP developed its own algorithm using a more improved strategy. So then secondary structure and then pro check, which are giving different scoring measures in a single server, and all of their limitations. Okay. So DSSP has been worked out by Quebec and Sender. So they wrote this script in 1983. So what their objective was, they were assigning the secondary structure. Understand that they were not at all predicting. They were only assigning the secondary structures. Okay, in the entire PDBs for all the PDB entries, and then they were building their own DSSP database, which is still there and still being extensively deployed. So how it is doing is understand the strategy is first it reads the protein structure, okay, and then it calculates all the diagonal angles, hydrogen bonds coming up, everything. So it reads the entire geometry and topology of the protein structure from N to C. Then what it does, it already discards all the hydrogen bonds. But what it believes is people are putting the assigning the hydrogen bonds in themselves, which may not be correct. So as to bring the uniformity in picture, everything is correctly done for all the PDB entries and the DSSP entries in their own DSSP database. They discard the additional bonds, okay, and then optimally place them. So now what is the strategy to place? What they are doing is they are placing the isomers at a distance of 1 Armstrong from backbone N and the carbonyl C double bond O. So this N and O, they are optimally placing the hydrogen atoms at a distance of 1 Armstrong without causing any clashes, without putting any strict hindrance on any other proximal atoms. With that constraint they are working, and they assign the partial charge as well. Like for carbonyl plus 0.42 E, and for amide is minus 0.2 electron. And then what they do, they calculate the overall energy, see, using this formula. 0.0841 upon RON plus 1 by RCH plus minus 1 by ROH minus 1 by RCN into 332 kilocalorie per mole. Then what they do is, when they do assign the part properly for all of them, then two best hydrogen bonds for each atoms are then used to determine the most likely class of secondary structure. Because every amino acid will be involved in several different hydrogen bonds. So what are the best two bonds, okay, which are having the lowest energy? So considering the best two bonds for each amino acid, they are assigning the secondary structure. Is it alpha helix, beta sheet? If what kind of alpha helix? Okay, what kind of helix it is? Okay, so this way the broken or incorrectly assigned structure can now be properly done. And how do you evaluate a prediction? Here, what they do, if the known structure was there, they assigned some structure. They assigned out of 100 amino acids, they were proper definition for 97. How much of the 97 amino acids are now correctly assigned the secondary structure which was already there, already found to be in the experimental crystal. Okay, so they compare the data using the Q quality score. Okay, see the data angles, the phi and psi concept or the omega concept. This we already discussed in the last class. N C alpha CO. This is the R side chain. The phi and the psi here. Between N and C alpha is psi phi. C and C alpha this plane is psi. It's rotating like this. And the omega is for the side chain. The glycine is omega is not there because it's hydrogen atom there. Now understand, see, if it is a beta sheet, they are assigning the character B to represent each amino acid in the beta sheet. If it goes normally, that any amino acid which is forming a beta sheet is B, uniform beta sheet. But if it is a beta bulge, some incorrect secondary structure is there, incorrect pattern of hydrogen bonding is coming up. Then what they do? They assign the extended strand, which is the E there. The B on the normal and E on the beta bulge. Remember that beta bulge is the part in the this beta sheet where most of the mutations also come up, just to absorb the mutations. Normally, what people say, like secondary structures are well conserved, mutations do not happen there. But remember that this is not true. Okay, show them the answer of beta bulge where many mutations still come up. See, this is the next part. See, you might have heard about the 310 helix, alpha helix, pi helix. What is the unique beauty here? Like 310 helix, I to I plus 3 bonding is there. You are maybe already knowing. Alpha helix I to I plus 4 bonding means one amino acid in forms a N. The common amino acid forms a bonding with the I to I plus 4. Fourth amino acid down the line. Okay, like that. And pi it is I to I plus 5. See? Then unordered structure in blue turn. See? So hydrogen bonded turn which is there. See? Because of hydrogen bonding, certain turn comes up in the secondary structures. See here. The next is the bend. Only because of hydrogen bonds, nature properly tweaks the secondary structure and connects them in a way to guide the function. But see, nature is such a beautiful artist. And see the coil here, see? Coil means any pattern can be taken, no consideration at all. Till now, like we have been discussing some strategic pattern, strategic conserved nature of some structure because of these hydrogen bonds, because they are properly sealing it up. But coil, nothing. Okay. They are not taking any of these states so far. So whenever no such state has been there like this, which we are discussing like G, H, I, T, S or B, E, we call it coil. Likewise, the scientists also do. Whenever they find any new unique part, they put that in coil. See the DSSP part. If you check up like this, you'll get the part like this. See? I mean, if it's secondary structure, solvent accessibility also turns up. What is the solvent accessibility part? Understand that solvent accessibility means if you put a protein structure in a solvent, how much like a solvent is able to assess the like intrinsic core, internal core. For every amino acid, like the outside surface area, okay, 4 pi r square because it's a like sphere. So 4 pi r square. Out of that 4 pi r square, how much surface area is being in contact with the water solvent? That is termed as solvent accessibility. That defines the solvent accessibility. So for an amino acid which is present on the top, means superficial, outside of the outside boundary, then that is like most of it is like 70 to 80% is outside. So solvent accessibility is 0.8. But something buried in the core, solvent accessibility may be 0. Okay. So by only calculating this on the for all the amino acids, I can get an idea like which amino acid is present in the core, which amino acid is present in the middle, not in the buried core, not in the outside superficial part, but somewhere in the middle and something on the outside, just by guessing the number. That is the beauty of solvent accessibility. See? Now ProCheck is the next program. Okay, it analyzes torsion angles, it analyzes bad contacts by which through van der Waal radius. It analyzes hydrogen bonding energy, perform overall and per residue analysis, it do many things. Okay, identifies distorted geometry as well. See? So ProCheck many people still use ProCheck. Okay. And for its extensive usage, you will find many such servers. It even includes Samson and map. See, only the distribution or pattern may be different. Like here, it allowed generously allowed. See, residues in most favorable regions. 83.3%. See, residue is additionally allowed regions. 13.1. Residues in generously allowed region. 3.6. Residues in disallowed region. 0. Giving rise to 100%. See, so I can say most 5 pairs should be in favored or allowed regions. If the Ramsden score goes high, the model is proper. Okay, but it is not the only sole criteria. Okay, understand that. So see, besides the this approach check, DSSP, like Ramsden map, different scores. People normally use structure comparison as well, which is currently being used extensively. Okay, so what we do here is that we compare our model structure against the solved structure, actual solved structure. If we already have the answer, and if you are participating in a test by predicting the sequence whose structure is already there with us, so we can always evaluate our modeling algorithm. However, uh, if we don't have the actual structure, then also we can compare our model structure against the template through which we are modeling the protein sequence. Okay, so what is the objective here is that once the model is built, we overlap structurally superimpose the protein model and the template or the actual answer, and then find out how many amino acids are exactly overlapped within a distance deviation of 1.5 Armstrongs. Means uh, 1 to 1 distance deviation is 1.5, less than 1.5 Armstrong. 2 to 2, 3 to 3, 4 to 4. Whichever amino acids have been modeled, like in the template which was taken as per the alignment, their alignment, the distance between individual distance between these two amino acids should be less than 1.5 Armstrong. Then that means the topology here in the template or the actual structure has been directly copied in the model as well. It's retained. Okay, so the model is correct for that aligned portion. However, if because of the insertion of gap or the linker region or the gap region was too big, it may deteriorate or like structurally displace the conserved segments which are taken from the template. Okay, so that may happen. So need to properly like pack the gaps as well by using refinements and extensively iteratively doing it. Okay, I'll show a few examples here. See this part, which I have personally modeled. See recently. My sequence length of like see 329 and 321 here. See 3FR9 I used it as template, and 3WH10A I tried to model it. So this was an extended segment which could not work here, and I did not try to fit in there. Although I could have done, but I did not do it. I like just made the alignment using modeler. Okay, was it OC? This was also there. Just to pack this part properly, I let it as such. Okay, so that the overall alignment can be properly worked out. Okay, so to make it that way, then I got the alignment as alignment is like this. This resulted a model whose overall RMSD was 0.633. See, the colorless is the template here, 3F5F9, and this cyan color, see, is the model exactly fitting. Here you are seeing some gaps here. See, this part is not overlapping, but I can understand. See, is it a good model or a bad model? As per definition so far, 0.633 over the overall RMSD is substantially very good overall topology. The displacement has only been coming there because of these loop regions. The secondary structures are well conserved here, see? Properly conserved. Now see, I want to evaluate here to continue our discussion how the length plays substantial role, how the template selection plays substantial role over the length. If I get the template with a significant similarity or not. As the length increases, we have been discussing that the chance of getting a substantially highly similar template decreases. Okay, so to understand that, I am playing a small experiment here. See, I am conducting a small experiment. See, I have taken two sequences. First sequence is length 346, other sequence length 207. I am calling that as sequence 1 and 2 with length 346 and 207. Now see the results. So see, for these two sequences, I am now taking two templates, like one substantially good one, one middle one. Here, the good template which was available was NA of 4QNN template with a sequence identity of 34.32%. I have done it using Swiss model. You can also do it for any sequence. The coverage like this, a very high coverage, more than 90%. The something which is having a lower sequence identity means, of course, coverage sequence coverage alignment length will be coverage will be much smaller. It will be the template will be able to cover or span a lesser number of amino acids. Okay, see, I'm getting a very low coverage only in the middle part, not the terminals. Now, when I built the model, I got Ramchandran's score of 87. Here I'm getting Ramchandran's score of 88. Is this model good? Is this model bad based on Ramchandran? Now you understand that Ramchandran only tells model is right or wrong. It does not tell how much the model is correctly built. Here the model is not at all built. See, I'm seeing the extended loops, like clumsy this, loops have gone inside the secondary structures. See, staggered part. But here the structure is well packed. See, some decent geometry. So as you have been discussing, when the sequence identity between target and template is going over 30%, the template based modeling, that is the homology modeling or threading, results in a substantially good model, like near native structure of a lower resolution NMR structure. Means the modeling is the homology modeling or the threading is able to build a model of low resolution NMR structure with that much high accuracy, as resulted by a low resolution NMR. Okay, NMR structure of that high accuracy modeling is able to build. So as the sequence similarity or sequence identity goes high, the model is very much high, very much accurate. See, you see substantial number of amino acids within the Ramchandran allowed region. See, only a few are not allowed. These highly allowed regions here green. but still 87.06. Clash score is 7.28. Here clash score is 10.84. As obvious. See, alignment is not at all proper there, nothing. 4.20 is Ramchandran outlier, something which Ramchandran favor, it should be in Ramchandran favor in this way, but it's not there. It's 5.32. See, rotamer outlier shows the different side chain is placed 2%, here 1.27%. Means such errors can be resolved by like proper side chain placement or rotamer correction. Okay, or rebuilding the side chain by using tools like Scroll, sidechain modeling with rotamer library SCWRL. You can check out. Okay. C beta deviation 5. C beta deviation is 8 means there is a substantial structural topology like a disorder is also there. See? But if you put sidechains properly, it can be corrected. Bad bonds 0 of 2287. Bad bonds 0 of 1474. See? Means model is very small compared to a very big model. Now the same sequence, they have a like substantially variant number of like bonds. No, even if the models are very close, they can have a little deviation, but not substantial deviation. So what we understood here, if the sequence identity or sequence similarity goes very less, the model is very bad, which cannot be even recovered by like a extended model sampling. Even if you do energy refinement for too much time or many cycles, nothing will happen. These scores will change substantially, but this model cannot be brought like this. Understand? We only need sequence identity or the good alignment. Okay. Both of them are there means very good model. See? Second, what is the take home message we got here? Even if the Ramchandran score is high, it does not impart the model is correct. We should also see the other constraints coming up in the picture. Because even if the Ramchandran score is high, it does not tell whether the alignment was properly there or all the amino acids properly covered with a high sequence similarity or identity with against the template or not. Further, it does not impart like tell any information regarding like whether the model was properly relaxed or not, like its energy is properly stabilized or not. But if it's a low energy model, Ramchandran score will always be high. Okay, no doubt about it. So Ramchandran score is just a parameter. Now see the next part. If I just consider, now see for the sequence 2, as the length is smaller, as we have been discussing so far, we can get very good templates very easily. See, not showing the sequence similarity case here, but see, Ramchandran favored 90%, clash score 5.4, Ramchandran favored 94.94%. So now you understand that which model is more accurate. See, everything coming properly there. See, here is like distorted structural topology. Here it is much more like properly packed structure. Okay, not much deviations coming up here. See, zero, zero. See, 664 and 839 are very close. So because if the many clashes are there, number of bonds may increase. So see, like it depends on exact inference how you are taking the picture. Okay, things should be properly analyzed, then only it makes sense. See, no score is good or bad. We need to properly understand. All these things should be properly considered in nature. Then only we can exactly model the things properly. Okay. So see, in the summary at the end, I would like to conclude that in this session we have studied regarding DSSP, Dictionary of Secondary Structure in Proteins, which uniformly place all the hydrogen bonds, properly assign the secondary structures, and resolve the errors that could have happened while determining the protein structures by some scientists, if so. Okay, this way we properly uniformly assign the secondary structures, keeping their definitions intact, so that by allowing the B, E, H, like T, C, we can properly understand each and every structure in every single algorithm. ProCheck, it assigns the evaluates the overall geometry of the protein structure, like bond length, bond angle, dihedral angle, the crashing atoms, okay, everything. So it evaluates the overall quality of a protein structure. Okay. So moreover, it also evaluates the Ramcharan favor residues, percentage, and outliers. Ramcharan and plot which you already seen. Okay. It gives the like a proper representation of distribution of the backbone diagonal angles phi and psi. Okay, and then it shows the how much like conformational space of the protein structure is well placed in the allowed regions. Okay, and how much is disallowed. Some percentage of by relaxing the threshold components, we also allow generously allowed. Okay, so the overall advantage is we can always evaluate the protein structure. Besides that, we have seen the utility of structural superimposition of the protein model over the actual solver answer if it is existing, or the template. Alright. Okay, so now at the end I will check how much we have understood so far. In the Ramcharan plot, which region represents the conformation allowed backbone diagonal angles for amino acid residues. You might have observed so far that only in the upper left quadrant we have been seeing most of the amino acids focus there, the backbone diagonal angles placed there. Okay, so what is the answer? Upper left region. Navigation in the Ramcharan plot is associated with the conformational angles of alpha helices. This is the lower left region. Because you need to remember some things here. Now let us resolve a few conceptual questions also at the end. Is hydrophobic core identified by the DSSP program? DSSP, that is Dictionary of Secondary Structure Proteins, it properly assigns the secondary structure uniformly in all the proteins, but it does not find the hydrophobic core. Okay, in the complete structure, what is hydrophobic core? It DSSP does not know. DSSP only assigns the secondary structure for each amino acid. So the answer is false. Which term describes the diagonal angles between the planes formed by four consecutive atoms in a molecule? What is the answer? The answer is torsion angle. Okay, but torsion is the correct term. Thank you for enjoying this session. I hope you might have enjoyed it. Best wishes.